So yeah, recording has been started. Okay, I forgot to record. I forgot to start. I'm starting now. So yeah, you can go through later also those videos. So as I was telling, today is a week 2 class. I'll be starting with assignment 1 week 1, which was last year's assignments. Okay. So do you have any doubt before that? You can let me know. You can just write down in the comment section, and I will just read it. Okay. So first question is, which of the following statements is oblique incorrect? First option, smallpox is caused by a double-stranded adenovirus. Smallpox claimed a lot of lives in Europe and was called as White Death. Plague disease caused an epidemic in Greece during 430 BC. So you have to say that which of the following statements are incorrect. So options are 1 only, 1 and 2, 1 and 3, 2 only, 1, 2, 3, 3 only, 2 and 3. So in the first week of lecture we discussed about smallpox, that it was a epidemic in like long back around 430 before Christ in Greece. So this is correct, but the other two are not correct. Okay, so options will be, which are incorrect, will be 1 and 2. Next question, which of the following statements are correct? Mechnikov hypothesized that cells were the major effector of immunity, which is correct. Koch postulated that the microorganism must be found in diseased but not healthy individuals. This is also correct. Exposure to cowpox can protect individuals from smallpox. This is also correct, because we saw that when the smallpox, like the cowpox pustule was getting inserted or injected into healthy man, he became protected against smallpox. The answer will be 1, 2 and 3. Which of the following statements are correct? The Gruber-Weidel test is used to diagnose diphtheria. Alexandra Yersin first isolated and characterized a toxin form Yersinia pestis. The bactericidal power of defibrinated blood against anthrax bacilli was destroyed by heating 52 degrees Celsius according to George Newton. So these are very memory based questions. Okay, if you go through those lectures from week 1, like if you go through those slides, you will get those informations. And so which is correct? So option is, the bactericidal power of defibrinated blood against anthrax bacilli was destroyed by heating to 55 degrees Celsius according to George Nuttall. So if you prefer, like if you have those, want to have those information, we can also go through previous week's classes. Those slides also I have uploaded. I think some of you might face having accessing those slides. I will just look into it, and we'll give you all access so that you can access those slides. So as we were discussing, the plague was like endemic around 430 BC. So as we are discussing, smallpox is a double-stranded DNA genome. Okay, and so we discussed this about variolation. Koch postulates we have discussed. White death, yeah, white death was a term for Mycobacterium tuberculosis. Okay, not for smallpox, it's tuberculosis. And then, yeah, this you need to just read and then remember. So this is the scientist name and what they have stated. So yeah. Yes, so bactericidal power of blood. What George Nuttall stated, defibrinated blood possessed a considerable bactericidal power against anthrax bacilli, and this power was destroyed by heating to 55 degrees Celsius. So if you hit defibrinated blood up to 55 degrees Celsius, the power will be destroyed. He came to find this. So we were where we were. So yeah, next question is, which of the following statement is are incorrect? The side chain theory of antibodies was proposed by Paul Ehrlich. Emil von Behring termed the serum as antitoxin against diphtheria. Claves and Loeffler first isolated Corynebacterium diphtheri. So which is incorrect? Okay, so the answer will be none of the statements. So we will just go through the list. So in this list you can see those some Nobel Prize winners name and what they discovered. From this list only the answer, like the question was asked. So which of the following is are correct? So you can see from week 1 specifically from this list and table they have asked questions. So you need to know that who has discovered what. So which is correct? Edward Buchner termed the substance in the bloodstream that has antibacterial activity as alexin. Edward Jenner first reported to the Royal Society on the oriental practice of variolation of smallpox. According to Koch, the microorganism must be cultured from the diseased individual. So this is true, and the first option is true. Edward Buchner termed the substance in the bloodstream that has antibacterial activity as alexin. Which of the following statements is are incorrect? In an allogenic transplant, the donor is an identical twin. Yeah, so yes. So we also discussed about allogenic transplant yeah, just a second. Yeah, we also discussed about allogeny. Yes, autologous, syngenic and allogenic. Allogenic means where those were twins, you know. Yeah, the donor is an identical twin. Sorry. So in allogenic it's not twin, it's a different entity, but in syngenic those can be identical twin. So the answer so the option allogenic transplant, the donor is an identical twin, which is wrong because it's not allogenic, it's syngenic. So it will be it's incorrect. And comparative immunology deals with immunological relationships between developing fetus and mother immunity. Systems biology focuses on the cells and molecules and how they respond to vaccines and infections. Yeah, so we can analyze, we can focus on cells and molecules and how they interact using system biology. But comparative immunology, let us see that, yeah, what is development and comparative immunology? So it deals with the analysis of shared or divergent facets of immunology among different species. Yes, so comparative immunology basically shares like how the factors of immunology or diverging facets of immunology among different species, how it's related among different species. Okay, that division is called comparative development and comparative immunology. Okay. Which of the following statements are incorrect? The persistence of antibodies upon vaccinia virus is longer than the influenza virus is true. We have read that for influenza I think it's 1 year, but for vaccinia virus it's more than 1 year. It's a longer period of time we will have the antibodies in your blood. Post-transcriptional regulation by non-coding RNAs is an example of tight regulation of immunity at the cellular level. No. Systems biology focuses on the cells and molecules and how they respond to vaccines and infections. Yeah, again this is true. So false is, or incorrect option is, post-transcriptional regulation by non-coding RNAs is an example of tight regulation of immunity at the cellular level. Which of the following statements are correct? Flagellin from gram-positive bacteria sensed by our immune system is an example of the diversity hallmark of the immune system. Immune cells, but not non-immune cells, express pattern recognition receptors. All the immune cells can express pathogen-associated molecular patterns. Okay, what are the options are correct? Flagellin from gram-positive bacteria sensed by our immune system is an example of diversity hallmark of immune system. No. Immune cells, but not non-immune cells, express pattern recognition receptors. All the immune cells can express pathogen-associated molecular patterns. No, this is also not correct. So, which are correct? It will be none of the above statements. Immune cells, but not non-immune cells, express pattern recognition receptor. No, it's not true. So we can go through where we talked about yes. So pattern recognition receptors, normal cells, non-immune cells as well as immune cells can have pattern recognition receptor. So it's not like that only immune cells will have those. And then pattern recognition receptors are recognized, like those receptors recognize pathogen associated molecular patterns. Okay, so yeah. Which of the following statements are incorrect? Option 1, mucosal associated lymphoid tissue is an example of the peripheral lymphoid organ. Bone marrow develops into a major lymphoid organ at 2 weeks gestation. Spleen is present in the upper chest location of the body. Okay, so which are incorrect? Mucosal associated lymphoid tissue is an example of peripheral lymphoid organ. Okay, bone marrow develops into a major lymphoid organ at 2 weeks gestation, which is not true. Spleen is present in the upper chest location, which is also not true. So these 2 and 3 options are incorrect. Pierce patches are predominantly located in the, which is incorrect option. Okay, let me see. The question is, Pierce patches are predominantly located in the large intestine. The interaction of antigen naive immune cells take place in thymus. Fetal pancreas is a major lymphoid organ in 3rd month after gestation. So the question has been, let me see. The question has been deleted somehow. Yeah, which of the following are incorrect? Okay. Which of the following are incorrect? So all three are incorrect. Okay, we will just go over those informations once. Yeah. So organs of immune system. We have read about lymphoid organs. Okay. So what is lymphoid organs? Lymphoid organs. This is the site for generation, maturation, and initiation of adaptive immune responses. Organized tissue in which lymphocytes interact with lymphoid and non-lymphoid cells and trapped antigen. So this is also organized tissue where lymphocytes interact with lymphoid, non-lymphoid cells and trapped antigen. Primary or central lymphoid organs, which are immunocompetent cells are generated and mature. Primary or central lymphoid organs, immunocompetent cells are generated and mature. Secondary or peripheral lymphoid organs, organs where interaction of antigen naive immune cells take place, present in path of lymphatic vessels, rich in lymphocytes and macrophages. So secondary lymphoid organs are where interaction of antigen and naive immune cells take place. Okay, and which is present in the path of lymphatic vessels and reach in lymphocytes and macrophages. So prenatal, what are the prenatal lymphoid organs or system? Embryo yolk sac, which is 1 week of development, fetal liver 3rd month, spleen 7th month, after birth bone marrow. Okay, so we discussed about thymus and cross section of thymus, bone marrow, spleen, okay, lymph node, lymphatic system. So yeah, so properties and hallmarks of immunity, you have to remember that what are the tide regulations. So at molecular level, tide regulation is transcription or transcriptional non-coding RNA regulators. Translational level, positive or negative feedback system, pro or anti secretions. Cellular level, suppressor cells, example M1 or M2 macrophage or T regulatory cells. And another properties or hallmarks of immunity, discrimination among self, non-self and modified self. Modified cell means it's multilayer or uninhibited cancer cells. Okay, we'll again go back to the question. Lymphatic system, okay, so okay, so we have yes, so we have discussed so far week 1 assignment. So here every options are incorrect. The interaction of antigen and immune cells doesn't take place in thymus, it takes place in secondary lymphoid organ. Okay, and fetal pancreas is a major lymphoid organ in 3 months of gestation. This is also not correct, and Pierce patches is also not correct. Next week's okay, so this is week 2 question and answer which was been asked previous year. Okay, so the first question is which one of the following statements is are correct? Splenectomy can result in the systematic infection caused by Neisseria meningitis. Which are correct? Okay, the bursus fabricius is not a lymphoid organ in humans. Is not a lymphoid organ. Okay, this statement is correct. Is a organ of birds. The bursus fabricius is a central lymphoid organ in chicken. Yeah, this is true. So options 1, 2 and 3 both are like every options are correct. Which one of the following statements are incorrect? The size of the thymus is higher in adults, which is incorrect. The size of the thymus is higher in the case of like infants. Okay, thymus only produce T cells but also leads to killing of T cells. No, this is also incorrect. Pro B cell express IgG, which is correct. So options, pro B cells express IgG, which is not correct. Okay, pro B cells that is prior to mature B cells expresses IgG, which is not correct. So options answers will be option 1 and option 2 are incorrect. So the thymus only produce T cells but also leads to killing of T cells, which is correct, but the answer won't be this because it asked for incorrect statements. Which of the following statements are correct? Appendix is a major site for B cell maturation in rabbit. Thymectomy is associated with an increase in the number of T cells. No, this is not correct. With the advancement in age, bone marrow is mainly filled with fat. This is also correct. So appendix is the major site for rabbit for B cell maturation. This is correct. So 1 and 3 are correct. Which one of the following statements are incorrect? Appendix is the major site for B cell maturation in rabbits. Thymectomy is associated with an increase in the number of T cell, which is incorrect. And with the advancement okay, this is the same question is repeated basically, like the options are same but it's correct and incorrect. So yeah, the which is incorrect, thymectomy is associated with an increase in number of T cells. Which of the following statements are correct? Mucosal membrane can aid in the entry of pathogens. Yeah, true. Lymph only moves in one direction. Yeah, we have so yes, we have discussed about this, that there are like arteries and veins, and lymph like passes through it, like in between arteries and veins, and there are lymphatic vessels. Okay, so lymph only can only moves in one direction. We will discuss about this in detail. Interstitial fluid either go back or becomes lymph. This is also true. Yeah. So it's called interstitial fluid in between arteries and veins, and then it joins with lymphatic vessels, and where you will see that it becomes lymph or lymphatic fluids. Which of the following statements are incorrect? 950 rats of X-ray can completely destroy hematopoietic stem cells in mice. Zygote is an example of pluripotent stem cell. Pierce patches are examples of peripheral lymphoid organs. Which is incorrect? Incorrect is zygote is an example of pluripotent stem cells. Sorry. Which of the following statements are correct? Hematopoietic stem cells are examples of multipotent stem cells. This is correct. Germline stem cells that only produce sperms are examples of totipotent stem cells. No, we will talk about this potency and then multipotent, totipotent, pluripotent. later. So transgenic mouse models can be used to investigate the immune defense against bacterial infection. Yes, this is correct. So which of the following is incorrect? Conditional knockout mice bear the deletion of the gene of interest permanently from also this portions we have not discussed yet. We will talk about this in today's class or next class. So conditional knockout mice bear the deletion of the gene of interest permanently from all the cells of the body. Pluripotent stem cells can lead to generation of placenta. A person can't survive if his or her spleen is removed. So all those options are incorrect. Which of the following statements are correct? Herpes simplex virus is an RNA virus. No. So Cre recombinase is originally derived from the bacteriophage, and tyrosine kinase from the herpes simplex virus is used as a negative selection marker during the generation of transgenic mice. So the correct option is Cre recombinase is originally derived from the bacteriophage. Which of the following statements are incorrect? G418 is an antibiotic that can inhibit the protein synthesis of eukaryotic cells but not prokaryotic cells. CD34 is a marker for hematopoietic stem cells. Western blot techniques is used to detect both the proteins and RNA. Okay, so incorrect is the option 1. G418 is an antibiotic that can inhibit the protein synthesis of eukaryotic cells but not prokaryotic cells. This is incorrect. And the another incorrect is Western blot is a technique to detect protein but not RNA. Okay, so options 3 is as well a incorrect option. So we will discuss about hematopoiesis. So what is hematopoiesis? It is generation and development of different blood cells. So when we will have embryo yolk sac, which is in 1 week of development, okay, then fetal liver. Okay, so you need to know that where this hematopoiesis happens. Hematopoiesis means where generation and development of different blood cells happens. So it like when babies or human beings are in developing state, at 1 week of development, the embryo yolk sac is a source or is a destination or the source of generation and development of different blood cells. When a baby is in its 3rd month of maturation, then it will be fetal liver. Spleen, 7th month after birth, it will be bone marrow where generation and development of different blood cells take place. So hematopoietic stem cells are few in number, that is 1 in 50,000 in bone marrow. In general, it is very less in number. So hematopoietic stem cells marker is CD34, or it's also named as SCA1. So you need to remember this, that hematopoietic stem cell marker is CD34. Hematopoietic stem cell in mouse can be completely destroyed by 950 radiations of X-ray. These mice can die in approximately a week time if they are not rescued by 10,000 to 1 lakh hematopoietic stem cells from syngeneic mice, which is 0.01 to 0.1% cells. So this is a transfer experiment to investigate a particular cell type play a crucial role in defense system against virus infections. So this is a group of syngeneic mice. One group is infected with virus, isolate the discovered cells. Okay. Another group was irradiated, transferred discovered cells after infection. Another group irradiate, transferred discovered cells after without infection. Okay, so we have seen that these cells were protected. So we have seen that after we transferred transfer discovered cells after infection, these groups were protected. Okay, after irradiation. So we will know about different cell type. So what is totipotent cells? Totipotent cells give rise to entire organism which is fertilized egg or zygote. Okay, so zygote is not a pluripotent cell, rather zygote is a totipotent cell. So totipotent cell means it has a efficiency or potency to give rise or to give rise or to make the entire organism. And what is pluripotent cells? Most cell types are pluripotent. Example, human pluripotent stem cells can give rise to all of the cells of the body but cannot generate a placenta. Okay, so human pluripotent stem cell cannot generate a placenta. ES cells isolated from embryos. Unipotent cells differentiate into only one cell type. Multipotent cells differentiate into limited number of different cell types. Pluripotent cells, most cell types, human pluripotent stem cells can give rise to all of the cells of the body but cannot generate a placenta. ES cells isolated from embryos. So pluripotent cells হচ্ছে যে most cell types, human pluripotent stem cells can give rise to all of the cells of the body but cannot generate a placenta. So why transgenic mouse is needed? So to investigate the role of signaling molecule in physiological conditions. So wild type means where no genes are knocked out. Okay, and this is knockout means some important genes are being knocked out to see what is the function of those genes. Okay, and then virus, you will infect with virus. This will survive or survive longer, and this round, like where you have knocked out or dysregulated those genes which are important in fighting against virus, will so what will happen after virus infection? Those groups of mice will not survive or die early. Then you have to investigate what are the antiviral factors, and you have to investigate viral load as well. So you will infect those with different virus, like different virus infection you will infect, and then you will analyze the number of cells through flow cytometric analysis. So these are the steps for generation of transgenic mice. So it is beautifully explained by professor in lectures. So introduction about mouse ES cells. Preparation of targeting vector. So this is a fertilized egg, 2 cell stage, 4. So this is dividing 4 cell stage, 8, 16 and blastocytes. This is wild type allele. This is a targeting vector, and this is mutated allele, where you have seen that some part of the gene is like some part of the gene is been mutated. So neomycin resistance gene, positive selection marker. Okay, so yeah, so these are the steps of generation of transgenic mice. After that, you will do introduction of targeting vector and screening, and you will generate chimeric mice and pure transgenic mice. So it is mouse, blastocyst, then foster mother, chimeric mouse, and then you will get pure mice. So, and also you can do you can apply like Krylok system to cut some gene, and then adding at some like deletion and insertion you can do. So this is a Krylok system. Okay. So so conditional transgenic mice, conditional mutation, Krylok system, I will come to that part again. Let us discuss the basics of blood cells, what kind of blood cells we have and what are the function. And so we know that we have monocytes, lymphocytes, neutrophils, eosinophils, basophils. So these are WBCs basically, white blood cells. and we have erythrocyte and platelets as well. Okay, so we know that cells are getting developed from hematopoietic stem cells, and hematopoietic stem cells are self renewing because it's a stem cell. So from that we get myeloid progenitor and lymphoid progenitor. From myeloid progenitor, dendritic cells are getting derived, and from lymphoid progenitor, natural key cells, natural killer cells, and then T cell progenitor, B cell progenitor are getting derived, and as well as dendritic cells are also getting derived. So, from T cell progenitor, we get T helper cell as well as T cytotoxic cells, and from B cell progenitor, we get B cells. So, these cells are called agranulocytes because they don't have any granules inside them. And from myeloid progenitor, we get dendritic cells, we get granulocyte, monocyte progenitor, eosinophil progenitor, basophil progenitor, megakaryocyte, erythroid progenitor. From erythroid progenitor, we get erythrocyte. From megakaryocyte, we get platelets. Basophil, we get basophil. Eosinophil progenitor, we get eosinophil. From granulocyte, monocyte progenitor, we get neutrophils. And also we get monocyte. And those monocytes get differentiated into macrophages. So this monocyte and macrophages are called agranulocytes as well, but this neutrophils, eosinophils and basophils, as they are having granules in their cytoplasms, these are called granulocytes. Okay, so we can also isolate immune cells from the like we can perform different immunology experiment and can isolate immune cells. Peripheral blood mononuclear cells. It has lymphocytes 70 to 90%, monocytes 10 to 30%, and dendritic cells. So we can do density gradient centrifugation. Separates by using Histopac 1077, which contains polysucrose and sodium diatriazoate. Okay, so here before centrifugation you will add this Histopac. Okay, Histopac you will add, and then above it you will add blood, and you will keep for centrifugation. Then what you will found at the very bottom, you will get red blood cells. After that you will have Ficol Histopac, that the cushion you have used. Before that you will get Buffy or PBMCs, and then you will get plasma. So after centrifuge, plasma layer 55%, which will consist thrombocytes, which diameter will be 2 to 4 microns, cytokines, hormones, electrolytes. Density of cells and components in this portion will be less than 1.077 gram per ml. Then you will get mononuclear cells, lymphocytes diameter 5 to 10 microns or medium types lymphocytes. Mononuclear cells also will be monocytes, which is also diameter 16 to 25 microns. Then there will be a filter disc, which is lymphoprep solution density. Okay, with the cushion you use. Then you will get RBCs. Okay, RBC, this layer will contain polymorphonuclear cells, which is eosinophil and neutrophil and erythrocytes or RBCs. So density will be more than 1.077 gram per ml. Isolation of immune cells in clinical stages. Okay, so you can in different clinical cell stages you can isolate those immune cells. So what is leukapheresis? The method is called leukapheresis. So we'll talk about granulocytes, which is a category of white blood cells which is characterized by granular cytoplasm. Example: neutrophil, basophil, eosinophil. Neutrophils and eosinophils can phagocytize pathogens. Neutrophils or polymorphonuclear cells, most abundant white blood cells, okay, which is predominant cells in pus, accounts for its whitish appearance, respond quickly following tissue injury, hallmark of acute inflammation. Granulated cytoplasm stained with both acidic, which is eosin negative charged, and basic dyes. Okay, that's why it's called yes. So whose cytoplasm is granulated, and also it gets stained by acidic and basic, so it's called neutrophil. Neutro means neutral. So for acidic stain we use eosin, which is negatively charged, and for basic stain we use methylene blue, which is positively charged. The granules are antibiotic in nature and contain defensin, spherocidins, cathelicidins, bacterial permeability inducing protein, hydrolysis, lysozyme. 50 to 7% of WBC is neutrophils, multi-lobed nucleus, so its nucleus is around 3, it contains multi-lobed, you can see 3 lobed here. Professional, highly phagocytic in nature, short lifespan 2 to 3 days, expresses pathogen recognition receptors or pattern recognition receptor. Produce reactive oxygen species. Leukocytosis and diabetes increases in neutrophils in circulation of infections. So, okay, the term is called leukocytosis or diabetes. So, whenever any infection happens, the amount of neutrophils gets increased in circulation. Defect in ROS production result to lymphadenitis and may cause chronic granulomatous disease or CGD, the primary immunodeficiency disease. Lymph node enlargement due to infection or inflammation of lymph node. CGD are pediatric patient and are highly susceptible to frequent and sometimes life-threatening bacterial and fungal infections. The infection is recurrent. Genetic defect in NADPH oxidase. Genes are located on autosomal chromosome. Okay, so capital A will be healthy, capital A small a will be carrier, and small a which is in recessive condition will be diseased. Simple test using nitro-blue tetrazolium test converted in blue colored products suggest NADPH oxidase are normal. If not, then indicate defective NADPH oxidase. So this is NBT or nitro-blue tetrazolium. If you add NADPH oxidase, it will be colored compound, and then in normal individual you will be able to see, which is NETosis, neutrophil extracellular trap. So NETosis is basically a neutrophil extracellular trap which induced by ROS, which is reactive oxygen species. So it's induced by ROS, microsize, microbial virulence factor, and cytokines, composed of chromatin and neutrophil granular proteins, and provide high local concentration of antimicrobial protein or peptide. Eosinophil, 2 to 5% or less of WBC. Okay, bilobed nucleus and stained by eosin, acidic dye, which is, that's why it's like negatively charged, acidic dye which is negatively charged. Phagocytic in nature, but this is not its primary function. Play role during allergic conditions and asthma. Play important role in parasitic infection. So the primary role is it plays important role in parasite infection. An increase in number and condition is called eosinophilia. Eosinophils, cytoplasm is crystalloid in nature and contain major basic protein. MPP consists of toxin for helminth, induce histamine from mast cells. Some protein can activate neutrophils and platelets, can cause bronchospasm, sudden constriction of smooth muscles in the walls of bronchioles. Eosinophil cationic protein, eosinophil derived neurotoxin. Basophils are non-phagocytic in nature. Least common granulocyte, when activated, release histamine and other inflammatory chemicals, which is rare in circulation, contain large granules filled with basophilic protein, and upon binding with IgG with Fc receptor, these cells release histamine. Histamine increases blood vessels permeability. Histamine also is required for smooth muscle activity. Cetirizine block H1 receptor and prevent acute respiratory distress syndromes, which is mast cell. Present in wide variety of tissues such as mucosal epithelium, tissues of digestive, respiratory, urogenital tract, skin and connective tissues. Muscles produce histamine, prostaglandin, leukotrienes, which increase permeability of blood cells and play important role in allergies. Basophils and mast cells cause type 1 hypersensitivity. Okay, so So type 1 hypersensitivity can be IgE mediated hypersensitivity. Antigen induces cross-linking of IgE bound to mast cells. So this is the mast cells where IgEs are present. So what happens? Allergen comes and makes a cross-link of to IgE. Okay, this is Fc receptor for IgE, and this is allergen specific IgE. So IgE interacts with Fc receptor presence on the mast cells, and then those are getting cross-linked by allergen. And so what happens? It induces cross-linking of IgE bound to mast cells and basophils, and also releases vasoactive mediators. So these are vasoactive mediators. Typical manifestations include systemic anaphylaxis and localized anaphylaxis such as hay fever, asthma, hives, food allergies, and eczema. So how the cells get activated? Allergen cross-linkage of cell-bound IgE. This is IgE Fc receptor. IgE is bound with Fc, and this is allergen. So it's mast cell. Antibody cross-linkage of IgE. So anti-isotype antibody. So antibody cross-linkage also can be happened. Option C, what can be chemical cross-linkage of IgE. Cross-linkage of IgE receptors by anti-receptor antibody, anti-idiotype antibody cross-link, then enhanced calcium plus 2 influx by ionophore that increases membrane permeability to calcium plus 2. And what happens? What is the outcome of this activation? What is the outcome of this activation? So this is degranulation is happening. These are allergen. So this is vasoactive amines. So this, it can react to smooth muscle cells, smooth blood vessels, mucous gland, blood platelets, sensory nerve endings, eosinophils. So degranulation result to change in morphology as well. So you can see under microscope that due to degranulations, when those vasoactive proteins are molecules are getting out, there will be change in morphology as well. So this is IgE receptors, high affinity IgE receptor A, and this is low affinity IgE receptor. So it has immunoglobulin like domains. This is extracellular space, this is cytoplasm. You have carboxyl end at like the C term will be inside cell, and and this is plasma membrane, and this is low affinity immunoglobulin receptors. Okay, so primary mediators of mast cells: histamine, heparin, serotonin, eosinophil chemotactic factors, neutrophil chemotactic factors, proteases. This increases vascular permeability in smooth muscle contraction, increased vascular permeability, smooth muscle contraction, eosinophil chemotactis. So these factors basically does eosinophil chemotaxis, neutrophil chemotaxis, bronchial mucus secretion, degradation of blood vessel basement membrane, and generation of complement split products. Secondary mediators: platelet activating factor, platelet aggregation and degranulations, contraction of pulmonary smooth muscles, leukotrienes, slow reactive substance of anaphylaxis, prostaglandins, bradykinin, cytokines. There are different cytokines: IL-2, interleukin 1, TNF alpha, interleukin 2, 3, 4, 5, 6, TGF beta. So systemic anaphylaxis, increased expression of CAMs on venular endothelial cells. Various effects are there. Drugs to prevent type 1 hypersensitivity: antihistamines, which take care of blocking H1 and H2 receptors on target cells. Chromoline sodium, which blocks calcium plus 2 influx into mast cells. Theophylline prolongs high CMP levels in mast cells by inhibiting phosphodiesterase, which cleaves CMP to 5' AMP. Epinephrine, adrenaline, stimulates CMP production by binding to beta adrenergic receptors on mast cells. Cortisone reduces histamine levels by blocking conversion of histidine to histamine and stimulates mast cell production of CMP. Yes, you can, if you have any doubt, you can just write down in the comment section. Yeah, you can, if you have any doubt, Gopinath, you can just write down in the comment section.
Can I speak, madam? If you don't mind, can I speak, madam? I am not able to type, so I am asking. If any pathogens are entering into the body, so how other cells are intact on that pathogen? Hello, ma'am?
Just give her a few minutes, she might come back. She might have gone just out, she will come right now.
Okay.
Mom, you have muted yourself, we cannot hear you.
Hello, yes.
There was a question from Gopinath, he might continue with asking it.
Okay, thank you so much, Elias. So, Gopinath, you can, if you have any doubt, please let me know. Like, you can comment down in that comment section. Okay.
Actually, I am not able to type here, so I want to speak. Can I speak?
Yeah, sure, tell me.
So, if any pathogen enter in our body, so how in our body other cells are interact to it?
How our body, how other cells are
Interact on it.
interact.
Ah,
Okay, so what happens? Pathogens can enter into our cells through different paths. Okay, it can be like airborne pathogens, can be from from your when you inhale, it can interact. Some pathogens from blood, if you have a cut or wound, it can interact. It can come. So, so as I was discussing about innate immune cells and adaptive immune cells, so whenever pathogens enters into our body, innate immune cells come into action. So what happens? We have our neutrophils, macrophages, dendritic cells uh, in our immune defense line. So those will identify that it's a foreign particle or foreign material, and then it will as I was discussing, pathogens will also have pathogen associated molecular patterns. Okay, so our immune cells or innate immune cells will then try to interact with those pathogen associated molecular patterns. So pattern recognition receptor will interact with PAMPs, and then will be able to understand this is a foreign particle or foreign material. Okay, so this is how it will interact. Then those like macrophages and all, then those will start releasing different factors, and those factors will induce to call other cells, other immune, other like cells which are associated with defense system. This will come, other phagocytosis cells will come, and then those pathogens will get phagocytosed. Okay,
okay.
so
Is there that pathogens enter in our body? After that is there chemical changes, biochemical changes happening in there?
happening inside body?
Huh?
Yeah, definitely. So as I was telling, different chemical factors like cytokines, interferons will get released. Okay, so as you can see, if pathogens, so if suppose there is a cut in your skin, and then pathogen has been get into it. So what will happen there? Every like WBCs will go, and then they will release different chemicals, and you will see that pus formation will be there. Okay, so morphological, like from physiological changes will be there. You can see if it's happening at outside, and if it's happening at inside, you won't be able to see, but yes, inside also there will be changes. So yeah, it's all biochemical reactions happens. Yes, you are right. So we were discussing about dendritic cells. So dendritic cells comes from a dendrite term comes from a Greek word dendron, which means tree branches. Okay, and something also called patrolling curve. So this cell was discovered by Ralph in around 1970s, and posthumously exception in Nobel Prize. He was awarded Nobel Prize in 2011. So originated from hematopoietic stem cells and derived from both myeloid and lymphoid lineage. So dendritic cell is the only cell which is derived from both myeloid and lymphoid lineage. Present in lymphoid and various immune organs, especially present in epidermal layer known as Langerhans cells. Langerhans cells were discovered by Paul Langerhans in 1868 and were mistaken as nerve cells of the epidermis. It is also present in gastrointestinal tract (GIT) and respiratory tract. Just a second. What? Okay, so it is also present in gastrointestinal tract, GIT, and respiratory tract tissues. So, dendritic dendritic cells having less hydrolytic enzymes compared to macrophages. Just a second. Having less hydrolytic enzymes compared to the macrophages, having few digestive bodies or lysosome, poor viability, and not so phagocytic compared to macrophages in vitro and in vivo. GM-CSF is used for in vitro generation of dendritic cells for various experiments. They take up antigen, induce innate immune responses, present antigen along with MHC2 to T cells. So these are the functions of dendritic cells, that they take up antigen and they induce innate immune responses, and then they present those antigens with the help of MHC2 to T cells. So dendritic cells instructing adaptive immunities. Plasmacytoid dendritic cells play important role during virus infection and induce robust amount of type 1 interference. Okay, as I was telling that those innate immune cells also produce interference. So we'll see that plasmacytoid dendritic cells will produce type 1 interference which help in antiviral state. Type 1 interference, interleukin 12, interleukin 18 will help in natural killer cell activation. These are all innate immune response. So these are, we'll talk about this in detail. And these are adaptive immune responses. So what happens, they with the help of MHCs, major histocompatibility complexes, they present those antigens to T cells. So what happens, they do antigen presentation which promotes effector T cell and Th1 cell responses, which drive T regulatory cell and Th17 cell responses. Natural killer cell crosstalk drive plasma cell responses. Immune regulation via induction of cell death and suppression of T cells, and immune cell recruitment. Okay. So follicular dendritic cells, these are specialized antigen presenting cells present in lymphoid follicles. Follicular dendritic cells function is to bind and retain antigens by linking to complement and immune complexes, and then present these antigens to germline center B cells that start the secondary immune response. Follicular dendritic cells aid in the rescue of bound B cells from apoptosis and induce the differentiation of B cells into long-term memory B cell clones and plasma cells. Macrophages. Diabetes is passage of blood cells through unruptured wall of blood vessels into surrounding tissue. The process is called diabetes. Activation of macrophage by pathogens or by cytokines. Monocytes and macrophages are originated from same lineage. Monocytes are small, less complex and less phagocytic, floating phagocytic cells. Macrophages are large, highly complex and more phagocytic in nature and present in different tissues. So this keep the system clean from cell debris, blood cells or blood clots or dead cells. Okay. Macrophage is based on tissue macrophage, named is different. In intestine it's called intestinal macrophage. So in lungs the macrophages are present, it's called alveolar macrophage. In a connective tissue, it's called histiocytes. In liver, it's called Kupffer cells. In kidney, mesenglial cells. In neural tissue, it's called microglial. And in bone, is called osteoclasts. Difference between macrophage and neutrophil. So macrophages are basically long-lived, months to years, present in tissues when it gets mature, able to migrate from tissues in lymph node, allowing antigen dissemination, not very strong stimulant for the inflammation. Oxygen dependent killing is not vigorous. Intracellular condition may permit the growth of intracellular microbe to grow inside. Pathogen can circumvent respiratory burst, break down phagosome, prevent phagolysosome formation, resist the release of granule contents. And neutrophils are short-lived, they are in circulation and keep circulating, not show such properties. Strong stimulant for the inflammation. Oxygen dependent killing is lethal to microbial pathogen. It is extremely hostile for intercellular microbe. Pathogen cannot if pathogen need to survive, then need to resist phagocytosis. Pathogen cannot circumvent respiratory burst, kill the neutrophils or get killed. So reactive oxygen and nitrogen species. So reactive nitrogen species are nitrogen oxide, nitrogen dioxide, peroxynitrite. And then reactive oxygen species are superoxide anion, hydrogen peroxide, hydroxyl radical, hypochlorite anion. So this is the reactive oxygen species generation reaction where NADPH helps. So we'll get to know about natural killer cells. So natural killer cells are 5 to 10% of blood lymphocytes. These are also granular. Granule contains perforin and proteases, induce pore formation in target cells and trigger apoptosis. NK cells have cytotoxic properties against tumor and virally infected cells through antibody-dependent cell-mediated cytotoxicity. Antibody-dependent cell-mediated cytotoxicity is mediated through Fc gamma receptor or CD16. So natural killer cells senses abnormal cells if target cells has reduction of MHC1, missing self or unusual profile of antigen MHC1. Natural killer cells can get activated by IFN gamma, IL-2, IL-12, IL-18. Chromium release assay, radio level chromium is used. Na2CrO4 introduced in the media and is converted to Na2Cr2O7. Natural killer T cells. So this is a subset of T lymphocytes. It shares both natural killer cells and T cells marker. Like the functions and share surface markers also. So what happens upon activation, the cells produce various immunomodulatory cytokines and influence other cell types. Immunomodulatory cytokines examples: Th1 cytokine, which is example is IFN gamma, Th2 cytokine, interleukin 4. Okay, plays important role against infectious agents, autoimmune disease, tumors, allergens. B cells. Prenatally, B cells are coming from fetal liver and later after birth it's generated from bone marrow. It is central to the adaptive immunity and responsible for the production of antigen specific various types of immunoglobulin. The function of B cells was discovered in the 1960s by Max Cooper. He demonstrated that antibody production was completely abrogated in chickens after surgical removal of bursa fabricis, which is the primary site of B cell development, okay, and subsequently irradiation. Therefore, the B alphabet from B cells is derived from bursa of fabricius. B cells are needed for CD4 plus T cell mediated responses, but it's not essential for CD8 plus T cells. So you can see that prenatally B cells are coming from fetal liver and after birth it is generated from bone marrow. Okay, and B cells is central to the adaptive immunity and responsible for production of antigen specific antibodies, okay, or which is also called immunoglobulin. So the function was discovered by Max Cooper and he demonstrated that in the case of birds or chickens, you can see that bursa of fabricius is the source of production of B cells. Okay. Cluster of differentiation. What is CD? So since these cells are functionally different, these express different surface proteins as well. So these surface proteins are cell surface receptors, signaling or addition molecule. Additionally, the same cells express different surface proteins during development process or during activation. Overall, these surface proteins are called cell surface marker. So overall, these surface proteins are called cell surface markers. Set of monoclonal antibodies specifically bind to specific surface proteins are known as cluster of differentiation. The cell surface protein to which this set of antibodies bind are called CD molecules or markers or antigen. The CD molecules has biological significance. It can be used for isolation or for characterization of particular cell types or even therapy. The CD marker can be used for diagnostic purposes also. The nomenclature of CD is done. CD along with number, it's represent order of discovery. The CD molecules are approximately 400 in number. So this is hematopoietic stem cells, and there you can see the development of different blood cells, natural killer cells, T cells, monocytes, neutrophils, like all WBCs, erythrocytes, megakaryocytes, platelets. So T cell, so the development of T cell and B cell we will cover in detail in next class. Here so here you can see proliferation and differentiation, native CD4 plus T cell. It can be differentiated into T helper cell 22, T helper 17, TH1, TH2, T regulatory cell, TH9, TFH. Okay, and different cytokines play important role in this differentiation. So this is B cell development and activation, which we will cover in next class. So yeah, so this is all for today, like the slides. If you have any question, we will start discussing, and all those parts like Krylov system I will talk about it in next class. I will prepare detailed slide and we'll discuss. And regarding different cells, T cell development, B cell development also we'll talk in next class. So in today's class, if you have any doubt, just let me know, we'll discuss about it.
Hello, ma'am.
Yes, tell me.
Uh, third week's study third week syllabus study material.
Sorry,
Uh,
tell me once again.
this third week study material and elaborate it.
Yeah, third week study material. So all the lecture materials are there in NPTEL portal. So if you go
Uh,
to NPTEL portal, you can see. I have. Am I sharing my screen? You can see my screen, right? So week one,
mm,
week two. So there are this this lecture materials. So where you can see those lecture materials, and problem solving sessions you can find here. Okay, you have to go to this problem
mm,
solving session recording, and you can find here.
mm. Uh, so same time after completing this class, or it may be there is a session part to that to third week syllabus general revision part.
Revision.
Revision at least revision like half an hour on that uh, third week topic.
Yes,
So it
yes.
will be help to quickly revise
Yeah.
sometime.
Sure. So today's class was for week 2, okay? So next class we will start
Uh,
with week 3. And if you want to revise anything specific from week 1 or week 2, just let me know. I will revise. Okay? I will revise at the beginning, and then we'll start with
mm.
week three's class.
That's
Yes.
like short note within a half an hour that whole week, but things should be covered.
Okay, okay. Sure, sure.
So as like memorizing purpose or it may be quick revision purpose.
Sure. Sure.
Hmm.
If you have any doubt, just let me know, okay? Or else we are done with today's class. We have discussed. We In the next class we'll discuss some other parts which I have not covered in today's class, and we'll start with week 3. And as I was telling, I will also quickly revise last class to you as well.
When our next week class will commence.
Our next week's class will be next week, Tuesday, 6 to 8 p.m.
Every
every week
okay.
Tuesday. Yeah.
Here is the end of this session of this class, madam.